there's a very important stem cell called a radial progenitor that gives rise to almost all the cellular elements, including the neurons in the developing brain. And these cells divide into more copies of themselves, or they can differentiate, which means they can become neurons. So we became interested in a protein called a kinase that has the abbreviation GSK3. We wanted to find a definitive way to understand what GSK3 does related to the developing brain. We were able to eliminate both GSK3 alpha and beta in a very early stage of mouse embryonic development. What we found is that in these embryonic mice, the entire head of the mutant mice was bigger than what we call the control animals. We might have hit upon a manipulation that increased the number of cellular elements in the brain. The overall hypothesis that we formulated was that without GSK3, the cell cannot respond to any of the signals that normally makes it differentiate. One of the most interesting implications of our work is that it provides a way that you might be able to manipulate the number of neurons in the developing brain fairly readily. We think that our demonstration now may turn out to be an important direction in schizophrenia research. Here is a GSK3 inhibitor that is in widespread use in the clinic. And this is the drug lithium that people may recognize as being important in the treatment of manic depressive disorder and mood disorders. One of the things that it inhibits is actually GSK3. And so we think that people will now need to think a little bit about whether lithium might have some unintended consequences related to progenitor division, particularly if the drug were given to children. Now, this is a long-term project, and we don't know whether this will work the way that we envision it.